All right. So thank you very much, everybody, for being here today and joining us for the presentation of Dr. Herbert Leonel de Matos Guedes. This presentation is part of IMUNNOWEBINARS conference series that is a joint endeavor of the Institute of Biomedical Sciences from University of São Paulo and the Carlos Chagas Filho Institute of Biophysics at the Federal University of Rio de Janeiro. Dr. Herbert Guedes graduated in pharmacy and obtained his master's and PhD from the Institute of Biophysics. He is currently an associate professor at the Institute of Microbiology at the Federal University of Rio de Janeiro and a public health researcher at Oswaldo Cruz Institute. After spending time as a postdoc at the University of Texas Medical Branch of Galveston, working in Dr. Lin Sung's lab, and at the Institute of Biophysics, working in Dr. Batira Bergman's lab. And Dr. Batira joined us today. It's very nice to see her here as well. His work has been focused mainly in the immune response to murine leishmaniasis and the development of vaccines. And as an expert in vaccines, he also joins the efforts to locally develop and produce a vaccine against SARS-CoV-2 infection. But his presentation today will focus on regulatory B cells and T cell exhaustion in murine cutaneous leishmaniasis. Thank you, Dr. Guedes, for this talk. We are looking forward to hear about your work, and the floor is yours.
Thank you so much for the invitation and for kind introduction. So it's an honor to me to be here to present in the institute that I did my graduation. So I hope that we can discuss part of my data that we're going to present today. And I also would like to thank Gustavo, Dr. Gustavo, for the invitation for part of the ICB from USP. So I'm going to share my screen.
Faz mês que eu escuto isso.
I think. Is it okay? So,
yeah, it's working fine.
Thank you. So leishmaniasis, there are two main forms. That's cutaneous and visceral leishmaniasis. So I have been working with Leishmania amazonensis that's responsible for cutaneous leishmaniasis and also diffuse cutaneous leishmaniasis. So usually people think that when you talk about Leishmania, it's like all Leishmania species are the same. So if you did it with Leishmania major, no, it's similar for all. It's not true. So when I look, for example, here, when it was performed the infection, comparing wild-type mice here in black with MHC2 knockout mice and HAG2 knockout mice, there are no lesions on these mice. So it's totally different when we compare with Leishmania major. If you look here, the wild-type mice, there's a control lesion when you're looking for a major infection, but when you look for HAG knockout mice, you can see here there's uncontrolled lesion. So that's totally different. That's observed on Leishmania amazonensis infection. So different parasites. So to understand more about the immunology of Leishmania, looking for Leishmania amazonensis infection, we can see here in the infection of interferon gamma knockout mice, we cannot see any difference. This work was performed for Batira Bergman's lab. So in the beginning of the infection, there is no difference, but in the chronic phase, there is uncontrolled lesion in interferon gamma knockout mice with increase of lesion progression and the alpha increase of parasite load. And for Dr. Ledo's lab, the same experiment was performed, and we can see here in the beginning of infection, no difference in the lesion progression, but in the chronic phase, we have a uncontrolled lesion and uncontrolled parasite load. So our group started working with B6 mice, and the difference that was demonstrated for professor assumed that just looking at the beginning of the infections, wait for more time. What you can see here, we have a progression lesion in the beginning of the infection until has like a peak. And then we have a control and you have a chronic phase that it's not like a major infection. You have a total control of the lesion progression. Here you don't have it. You still have the lesion, but it's chronic. It becomes chronic and stay chronic for way more than 1 year. And to prove that, you use mice from different sources. So we use mice from Unicamp, UFRJ, from Fiocruz, and from UF. And we can see that it's the same profile of infection. So we have a beginning infection, we have a peak, and then chronic phase. Here, for other cells, we have the beginning of infection, we have like a peak, and then we have a partial control and chronic phase. So it's similar, all the same. So we start to think that these infections are partially resistant mice. So we have the progression of disease and have a partial control. So that's totally different when you compare B6 mice from BCL mice that there is uncontrolled lesion. So diffuse cutaneous leishmaniasis, it's a problem because we don't have a DTH response, we have low IL-2 production, we have no specific antigen proliferation, and we have high numbers of parasites. So it's here. So if we compare like cutaneous versus diffuse cutaneous leishmaniasis, it's like we compare BCL and B6 mice, and BCL, it's like diffuse cutaneous leishmaniasis. You have the same phenotype on BCL mice. So this work here was performed by Dr. Claudia Brodsky's group. So she demonstrated that human dendritic cells, when infected by Leishmania amazonensis, we don't have any activation of human dendritic cells. So it was in 2007. And here from my group in 2019, we showed that we have no activation of murine dendritic cells, dependent of using white type and TLR9 knockout mice. What I want to talk to you and say to you now is usually when you study about Leishmania amazonensis infection, we just see down regulation. So, or you don't have activation, or you just have down modulation. So, decrease in molecules, decrease in cytokines, decrease in markers are not activated. So, we started to understand how our main question is how Leishmania amazonensis can inhibit effector responses. So what are the molecules associated to suppression and immunopathology? So those are our questions. And I don't want to be like surprised during the presentation. We are looking for IL-10 producing cells, focus on B cells, and also looking for PD-1 and PD-1. So we are going to present some of the data discussing these molecules. So we are looking at molecules that are upregulated that has capacity to downregulation effector molecules. So starting for B cells, so regulatory B cells producing regulatory cytokines, and they also has other markers like CTL4, LAG3, and the other molecules. So those cytokines has capacity to inhibit T helper 1, T helper 17, to inhibit the dendritic cells, monocytes, CD8 cytotoxic T cells, but also can upregulate regulatory cells, TR1, NKT cells. So these cells has like immunomodulatory phenotypes, inhibiting effector cells and stimulate regulatory cells. And it's something that still needs to be more understanding how B cells are induced to become regulatory B cells. And now you don't have like a marker that decides to be regulatory, so it is used as IL-10 producing. So if B cells are producing IL-10, we can consider like a regulatory B cells. All different types of B cells can be considered become IL-10 producing cells. So like B1 cells and B2 cells can produce IL-10, but there is a question about if some B cells can start So in the beginning of your life is starting to producing IL-10 and is still producing during his life or their life. So these are open questions about if there is or there isn't B10 cells. So in the beginning of the physiology of B cells. So it was demonstrated for Linson's group when you use GAD mice. So the mice that there isn't B cells. So you have a decrease of lesions without affect a parasite load in Leishmania amazonensis infections. So this phenotype was demonstrated before. And recently, not so recently, but in 2016, was demonstrated in Leishmania donovani infections in dogs, an increase of B cells producing IL-10. So in this time, we are working in the same time looking for these IL-10 producing B cells in Leishmania amazonensis infection. That's our questions. If Leishmania amazonensis is the induction of IL-10 producing B cells in Leishmania amazonensis infection. So we infected mice and we look in draining lymph nodes. What we can see here, we look in CD4, CD8, and CD19 cells. And what you could see, you have decrease of the frequency of CD4 and CD8 T cells because you have a huge expansion of B2 cells on draining lymph nodes. And we can see also in numbers, we don't have a difference. Of course, we have an expansion of CD4 and CD8, but you have a huge expansion of B2 cells on draining lymph nodes. And then when we look at the IL-10 produced, what we can see here, the major source of IL-10 on lymphocytes are come from B2 cells. So you can see here no difference in the frequency for CD4 and CD8, but you can see for B2 cells. And when you look for the numbers on the cells on the draining lymph nodes, we can see here the increase of IL-10 producing B cells on draining lymph nodes. So after that, we looked to see this phenotype in which mice could we use, but you need to have a B cells, but maybe you can have some problem to try to see if it changes some of the phenotypes. And we looked on XIG mice that there is mutation in brutal tyrosine kinase. This mice has low numbers of B1 cells, but it also has problem maturation and activation of B2 cells, and there is a problems of B2 cells interactions. So we infected XIG mice, and we could see that decrease of the lesion progression or delay in the lesion progression, but when we compare the difference in parasite load, we don't have any difference. Okay, so we have the difference and the lesion, but no difference in parasite load. So there is some role in inflammation based on the antibody production. So, but we couldn't now want to see what's the problem, what's the difference that these mice have in difference to the wild type. So first we're looking for the numbers of B cells in the draining lymph nodes. We can see here these mice. There was a decrease of B2 cells on the draining lymph nodes in naive mice, but we also have, after the infection, we have the expansion of the B2 cells, but we maintain the same phenotype comparing infected wild type versus infected ICB mice. We have decrease in the frequency of B2 cells and the frequency of the numbers of B2 cells. And you can see here the difference in the frequencies, it's high. So after we look for the antibody production, we can see here when we compare specific antibody production for against the total antigen of Leishmania amazonensis, we can see here that wild type mice has an increase or has more production of IgM production in comparison to XIG mice. The same for IgG. The difference is from IgG1 is very high, so you can see here wild type mice has more IgG1 in comparison to XIG mice. IgG2A and IgG2B have the same phenotype. So we have more antibody production wild type mice in comparison to XIG mice. So when we look at for in situ cytokine, we look for IL-10 productions. So we use the homogeneity of a foot pad, that the lesion, spleen, and lymph nodes. We also could see that there is a decrease in production of IL-10 in foot pad, spleen, and the lymph nodes. So when you look at using in the drained lymph nodes by flow, we could see that the IL-10 producing B cells in white type mice, but you cannot see in the XIG mice. There is abolished. So this production for B2 cells, also in frequency and numbers. So as we can see here, that XIG, there is no production, and the white type, we have production. So XIG mice has a problem with B1 cells. So we now know that there is a problem of IL-10 production when you're looking for B2, but you ask about B1 cells. So we started to see if the B1 cells can produce in vitro the IL-10 when it interacts with leishmania. So what we see here, when you did the culture of B1 cells from peritoneal and did with a simulation with LPS, we can feel an increase of IL-10 productions. But when you put bromocygotes from Leishmania amazonensis, we cannot see the increase of IL-10. But when we did put together LPS and Leishmania, you can see an increase in comparison to the controls at LPS. So the prime with LPS and after Leishmania, we increase the IL-10 production. So then we decided to infect the mice, and then to evaluate purified B1 cells from infected mice, and to look in vitro interact with bromocygotes and Xenica amachigotes. So we cannot see any difference in interaction if you compare B1 from infected and naive with medium spontaneous interaction with bromocygotes, but we could see an increase of IL-10 productions when interacting with Xenica amachigotes in comparison to naive mice. So prime the B1 cells from infected mice can produce IL-10 in interaction with Xenica amachigotes. So we decided to look the difference between naive and also infected mice to see what's the difference on B1 cells. So we infect the mice, and in the chronic phase, so we look at the peritoneal cells. What we could see here, if you look, this is naive mice who have B1A and B1B, but in the infected mice, we have decrease of B1A, decrease of CD5 expression. So it's like frequency. So if you have a reduction of B1A cells, we have an increase of B1B. So just remember that infection was performed in the foot pad. So this shows that the effect of the foot pad can change like the environment on peritoneal cells for infected mice. So it's a systemic change. So we decided to do the add back, so transfer B1 cells from XIG mice. That's a very good recipe. So when you add B1 cells, we can repopulate the XIG mice with B1 cells, but you cannot see any difference in the lesion progression. So it's very similar. And here we can see, so the infected mice, there is decrease in B1A population. The same phenotype was observed when we did the repopulation of the XIG mice. So XIG mice decrease B1A. Here we have the percentage on XIG mice, but in B1B we can feel an increase in infected wild type, and you have the same phenotype in the XID add back. So there is an increase of B1B population. So recently we performed the transfers of B2 cells for XIG mice, and we could see an increase of lesion progression more in this chronic phase. And now you try to understand what is this phenotype? Why is that the increase of this lesion progression? Is there more IL-10 or is there more antibody production? So still looking for to understand more this mechanism. So This work that I show here, it was produced during the PhD of Dr. Luan. Luan defended his PhD last week, and now he's on NIU, New York University, to do his postdoc. So he just moved for there. And so during his PhD, he had the opportunity to publish these two manuscripts. So our perspective about this work, we want to understand, so we show data on bulbous mice, but in the bulbous mice, we have the problem of effector T cells in these mice. So you want to see this phenotype of regulatory B cells in B6 mice, and you want to try to use the strategy to use the CD19 IL-10 flux just to abolish the IL-10 production on B cells. So it's our when. So we have in Brazil CD19, so we want to import to Brazil IL-10 flux to try to do this experiment. So now moving for the second part. So we saw the increase of IL-10 for B cells, and you see we have a lot of questions, but we start to look at the PD-L1 and PD-L1. So we know, so that is the PD-1 and PD-L1 are important pair that's involved in T cells APC interaction. So we can see here that T cells present PD-L1, that's interaction of PD-L1 and PD-L2 from APC cells. Now there are a lot of changes, so you can have the changes. So APC presents PD-L1 and T cells present PD-L1. Now it's more mixed, but traditionally. So you can see it's here. So T cells are expressing PD-L1 and APC is presenting PD-L1 or PD-L2. So when this happens, so we have the interaction of this pair, PD-1 and PD-L1. Of course, we have more markers for exhaustion. So like TIM3 and LAG3, and production of IL-10, TGF-beta, IL-2 reduction of TNF, reduction of interferon gamma, and then impair of CD8 and CD4 T cells proliferation. So this induces T cell exhaustion. So it was demonstrated for Alan Sharp groups. So a long time ago, in 2006, when infected PD-L1 and PD-L2 knockout mice with leishmaniasis infection. So here in the cycle, we can see it's the wild type. So when we infected PD-L1 knockout mice, we can see here, there is a decrease of the lesion and also decrease of the parasite load. And when we infected PD-L2 knockout mice, we have the increase of the lesion and also the increase of a parasite load. So PD-L1 and PD-L2 have two different roles, but PD-L1 it's associated to pathology because in the absence of PD-L1, we have decrease of the lesion and the parasite load. And was demonstrated in human with diffuse cutaneous leishmaniasis that it's ex vivo, but when you stimulate cells with the antigen, with total antigen, we have an increase of expression of PD-L1 by monocytes. So we started thinking, so at the same time, we're studying in murine mice model the expression of PD-L1. So this data was very important to us, to our findings. So we know that the treatment for leishmaniasis, there are several problems. The first line is the glucan. So there is toxicity, failure, and induction of resistance. So new therapeutic targets are necessary and urgent. So monoclonal antibodies could be one alternative to these treatments. So just to think about in the infection, the leishman infection. So when we have the infection, we have activation of these T cells, but the parasite is a chronic infection. So can have the ability to change the environment and to induce PD-L1 and PD-L1 on APCs and PD-L1 on T cells. The interactions between PD-1 and PD-L1 can inhibit or can induce exhaustion on T cells, reducing proliferations, cytokines, and cytotoxicity. Can you hear me? No?
Yeah.
Someone, yeah, someone muted it. Okay, sorry. So here, so in the infection, T cells can become exhausted because the ability of the parasites in the chronic infection. So for virus infections, this model are very established, and now it's moving more for parasitic infection like Plasmodium, Toxoplasma, and Leishmania. So we have the exhaustion of these T cells, but when you use monoclonal antibodies, so to block these interactions, we can revigorate these T cells, and these cells can induce and produce cytokines like interferon gamma, increases the toxicity, and to proliferate again. So this is very important to revigorate the immune response. So we started to try to see the presence of PD-L1 and PD-L1 cells. So we start in the draining lymph nodes. So we look at the draining lymph nodes for T cells expressing PD-L1 and the enriched cells expressing PD-L1. So we could see in the comparing infected and naive mice, we could see an increase of CD11C cells expressing PD-L1 in frequency and also in numbers. And also we could see in the infected mice, just remember that's Bcells mice, okay? We could see that increase of PD-L1 on CD4 T cells and also in CD8 T cells in frequency and also in numbers. So we have the increase of PD-L1 on the enriched cells and PD-L1 on T cells on draining lymph nodes. So we decided to start the treatment, so once a week, and we used anti-PD-L1, anti-PD-L1, and anti-PD-L2 by IP routes, so infected mice, 7 days start the treatment, and then we looked for parasite load. In this protocol, we could not see any difference in the lesion progression, and also no difference in parasite load. So we decided to move for treatment twice a week. So using the same treatment, so in the same protocol, but just treating twice a week. And what we could see, an increase of lesion progression on PD-L1 treatment and also anti-PD-L1 treatment. But at the same time that we have increase of the lesion progression, we have decrease of parasite load. So we're changing and simulating like an inflammatory response that, of course, we have an enhanced of lesion progression, but you control the parasite load. And we ask about the interferon gamma production. So we're looking for the treatment using anti-PD-1 and anti-PD-L1. What we could see also the decrease of parasite load in the draining lymph nodes and the spleen. So looking for T cells, we could not see difference in the frequency of CD8 T cells in those treatments and also numbers. And then when we look for interferon gamma production on CD8 T cells, we can see here, if you look naive and PBS, so there is no difference. Just to keep your attention here, when we infect the mice, we cannot see an increase based on the infection. You cannot see increase here in the frequency. So the infection on Bcells mice with leishmaniasis, there isn't increase of interferon gamma production cells. But when treated with anti-PD-1, we can see here a huge increase of frequency of interferon gamma cells producing. So looking here, in the treatment using anti-PD-1 and the treatment using anti-PD-1. So we could see an increase of in both treatment in frequency on CD8 T cells and also in numbers. And looking at the double staining for interferon gamma and for PD-1, we try to understand which cells are producing interferon gamma or becoming revigorated to produce interferon gamma. We could see here like cells that are positive for interferon gamma but negative for PD-1. We can think the treatment with anti-PD-1, there is an increase of this frequency here. the cells that did not produce interferon gamma but expressed PD-L1 will have a decrease in PD-1 treatment, just in frequency, only frequency, but not in numbers. But when you look here for the double positive cells, remember that are CD8 T cells that are producing interferon gamma and they also PD-1 positive, we can see an increase in the anti-PD-1 treatment. This data demonstrate that revigorating T cell response. So the cells that are expressing PD-L1 can also increase the production of interferon gamma. So when we move into CD4 T cells, we can see no difference in frequency and the numbers comparing the treatments. And when you're looking for the interferon gamma production, we can see the frequency for CD4, we have in both treatments increase the frequency, but only anti-PD-1 increase the numbers of CD4 interferon gamma producing cells. So we did the same protocol looking at double positive cells and in different subsets. We cannot see difference in interferon gamma positive and PD-1 negative for CD4 cells in frequency and numbers. And for PD-1 positive, interferon gamma negative, just as slightly increasing in anti-PD-1 treatment. But when you look at for the double positive, we have an increase of the frequency in both treatments, anti-PD-1, anti-PD-1 treatments, to increase the frequency of CD4 interferon gamma producing cells, but not in numbers. So we saw a reduction of IL-4 and TGF-beta in this in situ cytokine quantification, the footpad, but no difference in the IL-10. But this same phenotype was not observed for anti-PD-1 treatment, for example, when you're looking for IL-4. So after we're now interested into looking which cells are expressing PD-1 on the lesions, and we already know that there are a lot of neutrophils. So of course there is the macrophage, and the macrophage can express, but we start thinking maybe there are other players, and we think about neutrophils, and we start to look at that. So we're starting to recruiting these neutrophils using cousin model in the peritoneal cavity, and we performed the infection using leishmaniasis staining with CFC to be in green. And after 4 hours of interaction and infection, we're looking for PD-1 expression. And we cannot see it in the recruited neutrophils and then interaction ex vivo with leishmaniasis. No difference in the interactions with leishmaniasis, but we could see that for also promastogote and amastogote, an increase of PD-1 expression on these neutrophils. And when you look at like, because we have in the same interaction neutrophils that became green and neutrophils that are not green. So we cannot be infected because we didn't see. We cannot prove that there is inside or not. We know that the major parts are inside when you do a microscopy, but here we cannot say that. But when we have green neutrophils, we have more the induction of PD-L1. So the interaction is required to induction of PD-L1. And then we did it from bone marrow, and we purified neutrophils from bone marrow and did the interaction with leishmaniasis, and after 4 hours, we look for PD-1 expression. So what you could see that neutrophils from bone marrow are more infected or interacted more with mastogotes than promastogotes, and you will have a more expression of PD-1 expression on neutrophils from bone marrow when infected with mastogotes than promastogotes, of course, but both increase PD-1 expression in comparison to median. And again, just the green neutrophils that are induced, there is the induction of PD-1. After we perform the analysis using human neutrophils purified from blood, and then we did the interactions using the same protocol. And what we could see a mastigote has the capacity to escape the phagocytosis from human neutrophils, and you have more infected or more interacted neutrophils with promastigotes. But when we look at the expression of PD-L1, we just saw the induction when infected by a mastigotes in comparison to promastigotes. And the same phenotype, just the green neutrophils are expressing PD-L1. So we decided to look in vivo using murine mice model at BCLC. So you looked at 15 and 60 days after infection. So we infected in the ears to look in situ by flow cytometers, the presence of neutrophil, if there is or there isn't the induction of PD-L1 in these infected mice. So when you looked for 15 days, we can see an increase of the numbers of neutrophils, and we could see an increase of expression of PD-L1 expression, also in frequency and number, and the N5 of PD-L1 or ellipson-6G expression PD-L1 15 days post-infections. So when you look in the more chronic phase in 60 days post-infection, now we have a huge amount in the neutrophils and infect. So you compare here infected with naive, we have a huge amount of neutrophils in infected ears. And when you compare here is the frequency of neutrophils and the numbers of neutrophils, we have a huge increase of neutrophils of infected ears in the lesions. So then we could see an increase of PD-1 expression in infected, and also in numbers, and also in N5. So we have PD-1 expressing neutrophils on infected lesions. So with the help of Gustavo Menezes from UFMG, we perform in vivo experiments to do in vivo image to look at what's the environment using monoclonal antibodies, stained monoclonal antibodies to do this in vivo, and we could see neutrophils expressing PD-L1. So here we look for chronic infected mice, and if you look at the neutrophils in circulation, they're green, so they did not express PD-L1, that to be red, so to be orange. So in circulating neutrophils, there isn't an expression of PD-L1. But when you look in the lesion, of course, you have the lesion is a huge mass that you cannot see inside, but you can see in the neighborhood, we can see a lot of the orange cells. That's the combination of red, that's for PD-L1, and green for neutrophils. You can see here a lot of orange. And of course, you have a lot of red that are not orange. So there are other cells expressing PD-L1 that now we are studying, and I hope that as soon as possible we can start to dissecate more the difference of population of those different cells expressing PD-L1. But neutrophils express PD-L1 in the lesion. So after we saw the huge amount of neutrophils on infected ear, we think about the possibility of some of those neutrophils have been drained for lymph nodes. So we look in the lymph nodes in the chronic phase. So it's not used to see neutrophils in the lymph node, but we look at, and of course in the affected mice, we have a small frequency of neutrophils, but in the draining lymph nodes in comparison to naive mice. It's important to note that we don't do the perfusion of the mice, but you have this difference. And when you look in the frequency, of course, in the frequency number, we have an increase of the neutrophils in the lymph nodes in the affected mice. But it's surprising for us that when you look at the expression of PD-L1 on those neutrophils, we could see that there is an increase of the frequency of PD-L1 on neutrophils in frequency and number, and no difference in MFI. So those neutrophils that has been drained are expressed in PD-L1. So that's very interesting that maybe neutrophils can have a role in to interfere or to block not only the lesion, as we said before, but also in the draining lymph nodes. So we look for B6 mice because it's different BCL and for B6 mice, we also could see that neutrophils interacting with Leishmania, here it was promacygotes. So neutrophils from B6 mice after infection can increase the PD-L1 expression and also only in the fluorescent cells. And we perform the interactions between neutrophils and T cells. So what we did here is a culture of effector cells that it's developed for Professor Renata Meirelles, and we did the co-culture with CD8 effector cells with neutrophils. So here we did with naive neutrophils, but when we did the culture after infected neutrophils, we observed the reduction of interferon gamma production, and we treated using isotype antibody, we cannot see any difference. But when we did the co-culture blocking with anti-PD-1 or anti-PD-L1, we recover and also increase interferon gamma production. So neutrophils expressing PD-L1 can inhibit interferon gamma production for CD8 T cells. And we know that the most case of leishmaniasis is with Leishmania brasiliensis. So we think about let's perform in human neutrophils, and we stained, we purify human neutrophils from blood. We did interaction using our brasiliensis staining with CFC for hours after we looked for PD-L1 expression. What we could see that for using promacygotes and the amacygotes, axenic amacygotes, we can feel an increase of PD-L1 expression in human neutrophils for our brasiliensis infection, and also only in green cells we can see this increase. And we decided to look for humans lesions with cutaneous leishmaniasis to see the PD-L1 expression, and neutrophils express PD-L1 using immunofluorescence. So what you could see, an increase of double positive cells for elastasin, positive and PD-L1 positive expression cells, and here's the fold change. We have an increase of these cells. So we can see here this our collaboration with Professor Daniel Gomes from Federal University of Espírito Santo, Vitória. So this is the lesion. So in blue, DAPI, in green, elastase, in PD-L1 in red. So we could see here some orange of these interactions. So this work was performed for Dr. Alessandra during her PhD in my lab. And also here we can point to the collaboration with Professor Elvira. Moving now for B6 mice, because we have this difference. So we did a collaboration with Professor Sung, and to start to looking on B6 mice. Remember, I said before, that's totally different from BCL mice, present controlled lesion. So we are looking for in BCL. Now we move to looking on B6 mice. So we did the infection, and in the peak of infection, we start to do experiments. So first we did the real-time PCR comparing infected tissue and foot pad comparing to naive. We could see an increase of PD-L1 and PD-L1. These increases are not the same for other pairs, co-stimulatory pairs, like PD-L1 and PD-L1. So we're looking for draining lymph nodes. Here we could see on CD11C cells, we're looking for MHC2 and PD-L1. we could see an increase of PD-L1 expression on MHC2 positive cells on draining lymph nodes. So we could see increase of PD-L1 expression in frequency and numbers. And also we could see on CD4 T cells increase of frequency of PD-L1. So we have increase of frequency and numbers in CD4. So we did a similar protocol that we did in neutrophils, but now using bone marrow derived dendritic cells. So we infected with Leishmania amazonensis staining with CFSE. So, but now after 24 hours by flow, we look at CD11C, PD-L1, PD-L2 expression. So here we can compare dendritic cells that are not infected, so non-infected dendritic cells, the expression of PD-L1 and the expression of PD-L2, like basal expression. But when we did the infection, we have here again in the same interaction dendritic cells that are not infected or interacting, and here dendritic cells that are green dendritic cells that are infected or interacting. And here, looking these cells, so infected cells, we can see the expression, increase of expression of PD-L1 and reduction of expression of PD-L2. And looking the data, so for B6 and BALB-C, we have an increase of expression using a mash goats or promash goats of PD-L1 in these green cells, and decrease of PD-L2 in the same phenotype for BALB-C mice. And we start to look in the pathway. So we're looking for different pathways, and here for mTOR pathway, but just a point. So here we just look live cells using viability dye, and we also have the expression of PD-L1, but you're looking for the average comparing the infected versus non-infected cells treated or not. So here we use, for example, rapamycin to inhibit phosphorylation of mTOR pathway. And here you can see we reduce this average. So looking for different, like STAT3 and STAT5 inhibitors or inhibitors of Janus kinase and the others, MAC, and also we use MYD88 knockout mice. We could see that the most important pathway, it's here when we inhibit using rapamycin, warfarin, and STAT3 inhibitors. So looking for this average. So pyrethroid kinase, it's already been demonstrated that Leishmania amazonensis infection induce expression, and we're looking for induction of phosphorylation of mTOR on infected dendritic cells, and we could see here in the infected dendritic cells we have increase of phosphorylation of mTOR in comparison to when you're looking for no phosphorylated, and the controls, the infected dendritic cells has increased phosphorylation of mTOR. And to prove the impact of the expression of PD-L1 on dendritic cells, we compare bone marrow derived dendritic cells from wild type and from PD-L1 knockout mice. And sorry, we culture with naive T cells. We just purified CD4 positive and CD25 negative cells from spleen, and we perform a culture use anti-CD3 and IL-2, and after 5 days in culture, we perform flow. And it's interesting that you could see an increase of regulatory cells at a small increase, okay? And when you compare infected and non-infected, but in the PD-L1, no difference. But when you look at the expression of Tbet on these T cells, we could see a decrease in the expression of PD-L1 on T cells when we look at in the culture dendritic cells, infected dendritic cells cultured with T cells. But this difference was not observed and when you use PD-L1 knockout dendritic cells. So how about in vivo? So we look at the PD-L1 knockout mice to confirm that finding that we saw in vitro. So we could see that knockout mice has a decreased lesion here, and also decreased parasite load in PD-L1 knockout mice. And when looking for T cells, we could see an increase of Tbet and interferon gamma production on PD-L1 knockout mice and no difference in regulatory T cells on those mice. And looking for cytokinase productions, we could see in situ, right, in the lesions, we could see interferon gamma productions, increase of interferon gamma production in PD-L1 knockout mice, and decrease of IL-4, and dependence of decrease of IL-17. So we have the idea about in fact, the dendritic cells or Leishmania amazonensis can increase this pathway pyruvate kinase M4 and STAT3, induction of inducing PD-L1 on the dendritic cells that can inhibit T helper 1 cells. It could be one of the mechanisms of T cell exhaustion in Leishmania amazonensis. Of course, in the perspective, we want to looking for other molecules like CTL4, LAG3, and the other molecules. But to prove the role, the PD-L1 on the dendritic cells, we want to look at CDL1C3 and PD-L1 flox to prove the role of dendritic cells expressing PD-L1. So, what's time I still have? Sorry, just a second. I have more time, right? Yes.
Yeah,
we started. Yeah, we started 10 minutes later, right?
Okay,
I'm very close to finish. Sorry. So, and we're looking comparing Leishmania amazonensis and Leishmania brasiliensis. We have this difference that we have, like, of course, we have cutaneous leishmaniasis, but also when we have cutaneous leishmaniasis caused for Leishmania brasiliensis is different than when caused for Leishmania amazonensis. But we have the other pole that we have mucosal leishmaniasis that's usually caused for Leishmania brasiliensis here, that has DTH, has TH-ring response, has cytotoxicity, and has severe disease here. But when you're looking for self-healing cutaneous, it's different when it has Leishmania brasiliensis and Leishmania amazonensis. And for leishmaniasis fields, a lot of people are thinking about if there is PD-L1 induction in Leishmania brasiliensis infection, and if is there, why is there? You know, so these are a very good point. And this question was performed for groups of Professor Daniel Gomes that published this beautiful work that using lesions from patients, he demonstrated, and his group demonstrated induction of PD-L1 in CD4 cells and CD8 T cells. And when looking for PD-L1 expression comparing healthy and from cutaneous lesions, we can see more expression of PD-L1 and more expression of PD-L2. And you're looking in the immunofluorescence for CD58, an increase of PD-L1 and PD-L2 on macrophage here. So in patients, in tissues from infected patients, so then to try to understand if the inductions can interrupt or can block the interferon gamma or TNF production, he performed culture of cells from these patients, and his group performed culture using antigens, and in the culture using blocking antibodies against PD-L1 and PD-L2, what they could see here that there is increase of frequency of interferon gamma on CD4 and the CD8, and also for TNF alpha. And when you do in the supernatant of the culture, you could see an increase of interferon gamma in those cultures. So demonstrated that also in Leishmania brasiliensis infection, there is part of these cells that induce the PD-L1 expression and PD-L1, and can impair interferon gamma production. So my last moment that for B6 we have a lot of things to discover is just a small break that when you look TLR9 knockout mice infection, using Leishmania amazonensis, you could see an increase of the lesion. And here, we don't have a difference in the lesion, but you also have an increase in parasite load. And when you look in the peak of the infection, we have increase of the lesion and increase of parasite load. Here, of course, when you look at in the peak of the lesion, we quantify interferon gamma in situ by ELISA. We could see a decrease of interferon gamma in these knockout mice. But what I'm showing for you now is this data that when I look at interferon gamma production for CD4 and CD8 T cells, we just see problems in CD8 interferon gamma production in these knockout mice. And so we can feel in the white type, so CD8 T cells producing interferon gamma, but we cannot see in Thiele knockout mice, but you still have production in CD4 T cells. What I want to show for you this data now, because we still have a lot of basic questions on Leishmania amazonensis, the role of CD4 interferon gamma producing cells or CD8 interferon gamma producing cells, and what's the role of the induction of those checkpoint inhibitors in which moments, what's the role, if there is some functions. For example, we think about the possibility of these molecules associated to keep parasites and to maintain immunity, so we are trying to discuss that. So this work, just breaking the ideas here, is from Juliana and from Alessandra. So we still have a lot of more questions to try to understand the functions of IL-10 producing cells, with the role and the importance of PD-L1 and PD-L1. And we have a lot of work to do. So I need to say thank you to a lot of collaborators, and from Martira, from Elvira, André Vale, Célio, Susana, Renata, Alessandra, Patrícia, Fernanda from Federal University of Rio de Janeiro, from Fiocruz, Alda and Eduardo, from Federal University of Espírito Santo, Daniel Gomes, Federal University of Juiz de Fora, Elaine, from Federal University of Minas Gerais, Gustavo Menezes and Rodolfo, and for Dr. and Professor Linsons from UTMB, and for my institute, Institute of Microbiology, and the Instituto Oswaldo Cruz, and for the Institute of Biophysics Carlos Chagas Filho, and I have a straight collaboration with the Institute. I have been working for several years, and from different groups, and I'm also professor in the program of Biophysics Institute, so it's an honor to me. And I'd like to thank you to the groups that I have been working in Federal University, and the Oswaldo Cruz Foundation, and UFRJ, and Fiocruz. It's a pleasure to work together. This is the most recently pictures in my group. Now we have a recently joined undergraduate students, but I would like to thank to all my students and postdocs, because without them it's been impossible to do the experiments. And I'd like to thank the environment, that's a very scientific, positive environment with a lot of questions, very interesting students. Thank you so much for my students. This is a picture in the December of the last year when we started to have meetings, presential meetings, but outside the building to discuss the science. It is very good to return to the lab and just to discuss the science. And we did in the end of the year, Jardim Botânico, like to be outside the university and to walk and to have a good moment. So after 2 years to independence, we cannot do, but last year, in the end of the year, we did. It was very grateful to be together. Thanks so much for the attention.
Thank you very much, Dr. Herbert. It was very nice data sets, right? It's a lot of work, lots of students involved. I've been following your career and your scientific achievements, and I really wish to congratulate you for all you have achieved so far. And we usually open for questions from the students first. So if any students would like to ask a question, if they want, they could ask them in Portuguese or in English, whatever it's more comfortable.
Can I start?
Sure. Lucas. Lucas is my PhD student, is currently in Germany, and he's joining us from Germany. And as his custom, he'll be asking a question, right? Thank you very much,
Yeah.
Lucas. Yes. Please go ahead.
Thanks. It's 5 hours ahead here, so now it's 6 p.m. So good night or good afternoon for you. I have two questions. The first being regarding PD-1 in general, because I was wondering, I was reading about PD-1 and innate lymphoid cells in tumors, and I know that type 2 innate lymphoid cells express a lot of PD-1. So when you treat animals with PD-1 inhibitors, usually they proliferate more, so they can secrete more cytokines, especially GM-CSF, which can recruit and activate eosinophils and neutrophils. And what caught my attention was when I saw your neutrophil data and regarding PD-1 blockage and everything, I was wondering if maybe there couldn't be like a crosstalk between neutrophils and type 2 innate lymphoid cells, or type 1 like as in type 2 innate lymphoid cells that have transdifferentiated to type 1 like via plasticity. So if maybe there's an innate lymphoid cell response producing GM-CSF that could be activating those neutrophils. And also, just to connect, because my second question is specifically regarding neutrophils, the suppressor phenotype that you saw couldn't be related to maybe myeloid-derived suppressor cell phenotype, since they share several common markers with neutrophils. So maybe you could have a neutrophil response, but also MDSC response in this case.
Thanks so much, Lucas. Good to see you. So be careful with the water, okay? Please don't travel to Russia and to Ukraine now. Be in the Germany, please. Lucas, so thanks so much for the question. So I'm going to start for the second questions. So about the suppressor myeloid cells. So myeloid suppressor the markers is not so clear. So some people use just like ellipsis CG, ellipsis CC. For me, it's not enough to use as a major marker. So I have a lot of questions about that. But now, when we're looking first, we are more focused on neutrophils. But now, because we have a problem that I can share with you, we cannot see any markers of H480 cells. We cannot see in the lesion. So the marker for macrophage. But when you look using for FIG64, we can see it. So now use different markers, and we are comparing BALB-C and B6 to try to understand more, to try if you could see any myeloid suppressor cells, or it's only neutrophils or macrophages or monocytes. So we are now to try to understand this. This is how we are going now. It's Alessandra are doing these experiments now. So it's a very good question. We don't know. So we also know that macrophage pain expression, we now are discussing about the monocytes, so that are coming for the lesions. So I hope to answer this question soon. About the intraepithelial cells, we could see an increase of PD-1 in gamma delta T cells. So it's hard to look at T cells in the skin. It's not so easy. So we can say for you now, that we have the expression of PD-1 on gamma delta T cells, and we are trying to understand if this treatment can affect also gamma delta T cells. But what we can say that the PD-1 expressed gamma delta T cells, there's a problem in producing cytokines as interferon gamma and the IL-17. So we don't have the production of IL-4 in these cells. So we try several experiments, but we are trying other stimulus like antigens to try to see if you can feel IL-4 producing for gamma delta T cells. But now we just bought the antibodies to look in more cells, to look at IL-3s, and because you know that we need a lot of antibodies, so to discriminate. So I never bought so much antibodies to look at one cells, but we are doing now. So hope to answer for you in the future. You're muted, Bruno.
Bruno, seu som tá.
Yeah, the space bar did not work this time. Luisa Menezes, please. Thank you.
Professor Herbert, thank you for the talk, amazing data and amazing presentation. I have a question about the B-cell results with leishman infection. I'm very interested in B-cell physiology, especially in the subpopulations of B1 cells. And when you show the production of IL-10 by the B-cells in the draining lymph nodes, you show the general population of B-cells, B2020, CD19, but have you guys accessed the expression of CD5 in the cells? Because we know that B1A is just a tiny population in the blood and also in the draining lymph nodes and secondary lymphoid organs, but it has difference between these two subsets when you're treating of leishman infection.
That's a great question. So we did a panel to say like follicular B cells that are producing IL-10, and we look for CD5, and we didn't see the increase of this amount in the draining lymph nodes. But we have a question. We have a little bit, as because in naive we have, so we have the same defect, but we have a very interest in the lesion. So, but you have a point that I can share with you now, that it's a difference on B cells population when infecting in the ears and the foot pad. It's totally different. So it's a craving. So we expected to see a lot of B cells when it did the infection in the ears, but you have a huge amount of neutrophils. We have B cells, but it's not like when you saw in the foot pad. But the foot pad is so difficult because you need to remove the skin, because in ear, it's easy for us to open the ear and we can remove the tissue and not to macerate the skin. And you can put collagenase, of course, and you can look at the tissue. It's more easy for us. When you use the foot pad, it's so hard, but you have more B cells in the foot pad. So we're still wondering the best way. So we're trying to do in the ear that we have established to see tissue, but we still have to try to improve to look B cells on foot pads, because I think you have a more strong phenotype when looking B cells. So we have a question: if it is there B1 cells on the lesion? So because we have some expression of CD5 on B cells more that when you're looking for in the draining lymph nodes, when you look ear, but I think that if you look at foot pad, maybe it will be a more strong phenotype. Thank you for your question.
All right. So if there are any more questions from students, I would like to invite Dr. Marcelo Barcinski, that has raised his hand before. I guess he has a question. Dr. Barcinski?
Well, I haven't raised my hand, but I have a question, or I would say a suggestion.
Was feeling from Bruno.
Bruno is reading minds now. That's really good.
So you're a draft to pose a question. So it's your turn now, please. Thank you.
Oh, congratulations, Herbert, for such a nice talk and such a real amount of data that you have produced and that you have shown us. So I have what I would say a specific question. It is clear, as you said, that the dichotomy between Bcells and C57 blacks in resistance and susceptibility is not so straightforward for Leishmania amazonensis as it is for Leishmania major, but still it is the model that we have and the model that we must use. So my question is, and I think I wrote you as a suggestion when you asked me for suggestions on a paper that you were submitting, what is the possibility of studying F1s between Bcells and C57 blacks and see what is the genetic output of this of those mice? No? Are they purely resistant? Are they purely susceptible? Probably not. No? So you have the reagents and you have the competence and you have the personnel to see from a biochemical point of view with all the approaches, biochemical approaches and cellular approaches that you have, what is the phenomena that are dominant or they are co-dominant or that are recessive in those F1 mice. I think this is an approach that have never been taken. And I really don't know if an F1 mice from bulb and B10 is susceptible or resistant or intermediate between both. So following, as you show, the C57 black mice don't heal the lesion as they heal the lesion from Leishmania major. So at this situation where the lesion is not healed, is resistant, what is in an F1 black the phenomena, the biochemical and the cellular phenomena that are dominant, co-dominant, or susceptible? And a second point is that the difference between the size of the parasitophorus vacuoles between the lesion with Leishmania amazonensis and Leishmania major is really very striking. So the parasitophorus vacuole from Leishmania amazonensis infection is very huge and carry many Leishmania, very different from Leishmania major. So why not explore also why this parasitophorus, what is the mechanism or generation of such different parasitophorus vacuoles, and what does the consequences of this size, the dilution of the cytokines, and so on. So those are my comments, and again, congratulations. And I want to remind you that you are owing me one reagent that you promised to get. So this was just a joke. So again,
no, but it's true.
Congratulations again, and really I'm amazed with the amount of data that you and your group has produced.
So first, thanks so much, Professor Marcelo, for your question. Yes, I think we have to arrange the meeting to Professor Gerson to organize this. I'm so sorry to be so late because the amount of the experiment we're doing for COVID-19, it's crazy.
Yeah.
So, but so sorry for that, but I'm going to put you on. And first, it's we have in the Biophysical Institute in the program of immunobiology, it's we have a great opportunity to have the presence of Professor Marcelo Bacinski in this group. It's a great opportunity to discuss immunology, and also with Professor Julio, Professor Bartira, and the others. It's an environment so interesting with so many expertises and so many acknowledgements, and the experience of you, Professor Marcelo and Professor Julio, improve our outcome. So usually when before submit a manuscript, I can share with Professor Julie and Professor Marcelo, and they ask, what do you think about this manuscript? And the part of the result I show for Professor Marcelo and for Professor Julie. Thanks so much for your when I send it for you, you send me a lot of questions. So let's discuss about these questions now. And about the F1 mice, it's a very great idea. So we didn't start to do, which we know about a major infection. We have intermediate lesions. So for L major, and there is a lot of variations. So when you look for L major, like we have bulb C and B6 controlling, and for the F1 mice, you have a difference. So we can look at genetic background of this difference and to see why are they using biochemistry and now sequence. We can try to see why some mice that are more resistant and why some mice are more resistant. So these are very good questions. We need to consider to do that for this phenotype that you have for Leishmania amazonensis. So one thing that I can say about the difference that we saw for Professor Lin Sung's group and for other groups in the United States, because they don't have too much time, it's very expensive to maintain mice on bioterrium in the facility. So when we look at the lesions and they saw the difference, they kill mice. So they found difference, they kill mice. And usually in my lab, we did a protocol that first we look at the profile. So we're looking for a long time infection. So we have an experiment that we performed for 6 months. So usually people ask, why it's so long? Because we need to look at the all profile. So we bring from Brazil Leishmania amazonensis from Professor Lin Sung's group. So we performed effects with different species of Leishmania amazonensis, and our Leishmanias that we have, our strains, there are the same phenotype. So we are sure about those phenotypes. So this phenotype that we have, you know, the peak and then the resolution. So we can use the F1 as a strategy to understand. It's a very good suggestion. We promise that we are going to perform this experiment, Professor. You'll be my pleasure. And the second, it's about the innate of the parasitoid tuberculosis. It's a very good question. We all discuss this in our lab, the difference, because and now we have the non-2 knockout mice. We want to try to see some difference when looking, compare L major and L amazonensis, because there is difference in the infection of these mice. So we have a lot of cases, for example, for treatment. So when you treat mice with glucanthimine with L major and with L amazonensis, is there difference? Because when you look for bulb C and B6 mice for L amazonensis infection, you have a difference. We know that the immunological background of mice are important in treatment. So these are open questions. Recently, I did this question for Professor Van der Leyen. If someone proved if the big vacuoles, it's important for the chemotherapy. So I asked Van der Leyen like that. Professor Van der Leyen, did you know if someone tried to see if the importance of these big parasitoid vesicles? I asked him, he said no. So now you ask me, so we still, I think that nobody looking for this point, so we need to look. So we need the collaboration of a strong group in microscopy and to try to understand this difference in this parasitoid vesicles. So I think that's a very important point and question. Thanks so much.
Thank you very much.
All right. Does anybody have any further questions? Miriam, please.
Thank you, Bruno, but thank you very much, Herbert. It's amazing to see your work put together, and it's beautiful to know that you're doing that in parallel to the COVID-19 experiments. It's really beautiful, the work that you're doing with your lab and your collaborators, so congratulations on that. And my question has to do a lot with my ignorance on those types of lesions, and I was impressed by the images that you showed when you show the lesion stain for the neutrophils and the PD-L1. And I wasn't it wasn't clear to me, like you saw a lot of green, which meant, well, that you had a lot of neutrophils in the lesion, and I was wondering if they were basically covering it up or making the granulocyte, right, the granulous formation, or if maybe that was what that staining meant, in the sense that you did see some orange cells, like you could see the co-localization, like in principle, but not when the cell was attached to the lesion. So I was wondering if there would be subtypes of cells, of neutrophils, so the ones that are not yet forming the lesion, they do express PD-L1, and once they are part of the lesion, or if they have been there for a while, they stop expressing it, or if there might be some caveat of the technique. It wasn't clear to me why you did have, like, I was expecting to have it all orange, basically, or but that the difference that you saw between the cells that were composing the lesion versus the ones that were on the nearby was striking to me. I was wondering if you were able to investigate that.
Thanks so much, Miriam. So we still need to talk about memory cells. That's we need to look at that, you know that. So yes, a very good question. So it's not multiphoton. It's a fluorescence microscope. It's a confocal microscope. That's Gustavo Menezes' work. So we don't have the power to go much deeper, you know, in the lesion. So we have, it's a more beautiful image about the cells, but you don't have this capacity to go inside, to go deep in the lesion. So because that's we saw this being green. So we need to look in the neighborhood, you know, there's the decrease of amount of cells. So in the first moment before to do the experiment, I was thinking or wondering how we're going to look in walking cells, and it'd be so beautiful, but in the lesion we saw a lot of stuck cells. Oh, stop it, you know. And I was so surprised when we did some recently infections. So we could see neutrophils, you know, walking. It's so beautiful. We have some data in the beginning of infection, and the neutrophils are arriving in the tissues, but in this moment, the neutrophils are not expressing PD-L1. But in the chronic, it's stuck, and we could see a lot of expression of PD-L1. Of course, we have and neutrophils that are not expressing PD-L1, and you have a lot of cells that expressing PD-L1. So my opinion, I think we need to try to do more cut, you know, to try to do classic confocal, to see better, to do the reconstructions of the lesion. So if you could do like lesions into the sections, into the reconstructions, is a better way to us to completely understand the environment of the lesions. So we need to do that. So because in vivo imaging, there is not powerful to us to understand this. So it's one of our plans. So it's hard to do, right? To do these sections, and it's not easy. We are still learning how to do. So, but you want to do, you know, I think if you to do a 3D reconstructions, it'll be the better way to do several sections. So it'll be the better way to do that. It's in our plan to do. So, but it's not easy, but we are planning to do. And then with data that we have, we cannot say that, you know, we just doing more. We have some cells that this work is like to say we have neutrophil expressing PD-L1. It's something so new that in tumor cells, we are doing experiments since 2015, and it was published in 2019. The first paper about neutrophil PD-L1, neutrophil expressing PD-L1 was 2019 in tumors, but we are doing it since 2015. But we did so many experiments to prove that, you know, to not to be something quite, you know, and to be any problem. So to be sure, there is just few works in the world about neutrophils expressing PD-L1. So it's more common to see neutrophils as kamikaze cells, right? To do that and die, net. But we have other things that neutrophils can do regulations. Of course, it manipulates for infections or for environment, but we need to understand more about that. So using the expertise of Professor Elvira, because Alessandra was supervised by me and by Elvira, was easy when I do this type of collaboration. So we in mice model and the PD-L1, and Elvira neutrophils was a very good environment to discuss more. We have a lot of questions, so we want to do more about that for sure. Thank you so much for your question.
Thank you. Thank you.
I would like to apologize because I put a lot of data and I was deciding if you do one and to discuss more results, you know, and I'm so sorry for that. So Professor Julio always talked to me about that. My anxious to share all results, so I maybe it could be just for PD-L1, then B cells. So sorry for that, Professor.
Well, the problem is that you make us very frustrated, because we would like very much to be able to make some sort of intellectual contribution to the debate. Right. But I think that there is so much information in the presentation that we could stay maybe 1 week and we would still feel that it's too much, you know. So sorry. I think that maybe I could try to explore a little bit, you know, a specific comment that you made at the end of your talk, which may have a connection to what Marcelo has raised. Because finally, at the end of your talk, we had a scheme, you know, so the scheme we're missing. So at least from the standpoint of the dendritic cell biology, there is something that could be interesting to discuss, because I think that you mentioned that there is a very strong phenotype with a tau 9 knockout mice, right? Now, the question that comes to my mind is whether these large vacuoles containing so many parasites, are they also shown or seen in dendritic cells, or this is a particularity of macrophage biology? Because if you still have the same process happening in the dendritic cells, and you have the comparison with major, at least you could have a model, because then you could say, here I have, you know, vacuoles that are full of parasites as compared to dendritic cells that have few parasites. How the difference translate into the levels of expression of PD-1, let's say, or PD-1L, right? So you could maybe resume or condense, you know, this huge universe of information to specific questions that could be pertinent to what you're studying, because a regulation of immunity is extremely complex. I mean, you see Lucas Bolini, he likes innate lymphoid cells. So obviously he's going to bring another element to the circuit. So this is endless. I mean, it's endless. I mean, you can stay, you know, probably 1 year discussing how these components behave. Probably the orchestra, you know, has to balance itself. And, you know, several components are playing a role. There were a few questions that we always discuss in the past, which from my viewpoint, they are not resolved as well. For example, mastigotes, they should be the critical parasite components in terms of the regulation of the adaptive response. And you started the discussion talking about B cells, okay? So what is known about the repertoire of the BCRs if you compare mastigotes versus amastigotes, right? Because during the course of infection, these mice will not see promastigotes anymore. What they are seeing are amastigotes. So you have a lot of glycobiology there. You have a lot of sugars. I mean, I don't have any idea how the B cells are recognizing the MS signals. Is there any differential ability of the MS signals to stimulate via BCR? And in that term, you know, control the regulation, the expression of PD-1. I mean, you can ask the same question regarding the T cell component, because presumably the T cells are recognizing MS signals epitopes. They may be shared or not with the promastigotes, but you may have, you know, some specific epitopes that are being recognized, right? And that could contribute to your phenotypes. So I feel frustrated because I would like to be able to contribute more, but there are so many questions that are open that I cannot be more helpful than just to tell you how confused I can feel, you know. So this is my.
It's the feeling, right?
Yeah.
No, but no, you are contributing, Professor Julio. So starting for the dendritic cells, so you did a surgery question. So you have interesting to know how is the infection on the dendritic cells. It's for a long time because we are sure that it's different from macrophage. So we did perform some experiments, but you need to do electronic microscope to be sure, because when we do the infection with promastigotes and amastigotes are totally different. Totally. So, and we know that we have some important questions that can be making in this model. So we know that we have a more neutral pH on the dendritic cell than on macrophages. So what's the requirement to conversion of promastigotes and amastigotes? We have basic questions, but maybe I'm not going to answer these questions, but the immunological aspects, when we did the infection with amastigotes, axenic amastigotes, that we proved that they are very similar to the real amastigotes. And the promastigote, we have a totally different outcome on the dendritic cells using amastigotes. So we know already because Professor Sung's group, a lot of collaborations, works in collaboration with Professor Marcelo also. So we have some questions on that, and using the knockout can help us to better understand. But this I can be sure for you that the difference is clear. Using amastigote and promastigote, and we'll have questions, basic questions on the dendritic cells. And about this question about the repertoire, it's a very good question, and I agree with you that amastigote is more important for us than promastigote. So because that we put effort in our lab to do axenic amastigotes with all controls, because we have difference in the phenotypes. So one phenotype to discuss here, when you use the human neutrophils, axenic amastigote has ability to escape the phagocytosis. It's beautiful. It's something that me and Elvira and Alessandra was like, what? Why and how? Escape. Promastigote, it's all phagocyted by human neutrophils, but the mastigotes escape. It's something that we need to understand why. So the real lesions, we have the human neutrophils there, and the mastigote, and the mastigote maybe has escaping, and so we need to understand more about that. So there are basic questions, and of course to use more mastigotes than promastigotes in our ex vivo experiments it's quite important and try to do that. And repertoire, we don't know. We need to study more and to understand more and to do more collaboration with Andrea Valle. So we start to do some things, some together, some experiments to try to understand some points. About the glycobiology, we now using CD1D knockout mice to try to understand these recognitions with the impact of the absence of the CD1D for LPG. We know we have some phenotypes already, but LPG is from promastigote. So we now to start to collaborate to Rodrigo from Fiocruz Minas, René Rachu. We got some LPG from his lab, but we need more molecules from a mastigote. And we try to understand the impact of the absence of CD1D in vivo. That's our expertise, to do in vivo experiments to understand the immunological response. But now we need more molecules to try, because glycobiology or lipoglycobiology is quite important. We know that there is some molecules that act to produce IL-10 and to produce IL-17, for sure. And
so you don't know if the BCR is being engaged in the models, because you show so many data, you know, and it's not clear to me before the facts if you have stimulated with emastigals, with promastigals. It's very difficult to relate to the data, because I don't know if there is any component.
No, you just do
secondary response.
No, it was due to in specific stimulus in those experiments. Was PMA
and not specific.
Yes.
Okay. Now I get it.
Sorry, because we gave the manuscript for the students, I straight find the results. So sorry.
Anyway, I'm very impressed with your capacity to deal with so much information, because it's not easy. So you have to spend a lot of time studying your own data, for sure. It's good that the students are around.
Yes.
So they can help you.
No, but one point that sometimes is missing, and usually students like to think about your project, right? And usually I think about the project of the different students, and sometimes I do some like internal meetings to put the students to discuss the results. And I think something, I don't know if you remember, Professor Julio, that when I was doing my master, that Professor Baturi asked me to present my data in the program. So I think something that could be important sometimes, if some students want to share the data to discuss, you know, in our group, because when we have different people looking and different research with different experience, can help us, you know, to discuss a different point of view. I think that's something that's very helpful.
Okay, Dr. Guedes, thank you very much for your talk. Very insightful discussion. But I think we are a little beyond our time today. But it was very good discussion indeed. So thank you very much for everybody that stayed here with us so far. And thank you again, Herbert. It was really great to hear all this work, all this huge amount of data. You should be proud. You should be proud.
Thank you so much.
I know I am. To know you.
Thank you so much.
Márcio, você pode terminar a